Well, thank you. It's my great pleasure to introduce my friend Don Cleveland. Don grew up in New Mexico and went to New Mexico State University, where he got a Bachelor of Science degree in physics, and parenthetically also graduated as the valedictorian for the College of Arts and Sciences. He then moved eastward to Princeton, where he got a PhD in biomedical sciences, and ironically, he was in a class of 10 that included his fellow awardee tonight, Elaine Fuchs. He then did his postdoctoral work at UCSF with the great Bill Rutter. He joined the faculty at Johns Hopkins in 1981 before moving to UCSD in 1995, where he became the chairman, ultimately, of the Department of both Cellular and Molecular Medicine. Now, Don's actually made contributions in two fairly distinct areas of biology and medicine. His contributions, frankly, neither one of these areas would already make him one of the most impactful scientists of his generation. So, for example, he studied how chromosomes and genes get missegregated when cancer cells divide, and he's worked out the underlying biochemistry as well as taught us some of the therapeutic implications. But in addition, he's made seminal discoveries related to ALS, the dreaded disease, which is frequently better known as Lou Gehrig's disease. And by studying rare families that develop ALS on a genetic basis, he made completely unanticipated discoveries related to the root cause of ALS, including the unexpected involvement of both neuronal and neuronal cells in this disease. And this has led to ongoing clinical trials with new therapeutics. So not surprisingly, Don has been elected to the National Academy of Medicine and the National Academy of Sciences. He's won multiple prizes, including the Breakthrough Prize in 2018. For those of you who don't know, this prize comes with a very nice check. It's a little bit larger than the check you'll be getting tonight. All kidding aside, it's $3 million. And Don turned around and donated it to 3 charities, which I think is quite remarkable. In fact, even that In fact, if you're wondering, I took some notes because you may want to try to, it's the Doctors Without Borders, the ACLU Foundation, and the World Central Kitchen. So I think for all of these reasons, I can't think of a more worthy recipient of the Hopes Fund Awards this evening. So Don, thank you and welcome.
Well, what an honor it is to receive this award from the Hope Fund. It's now well known that genome instability is a hallmark, including complex genome rearrangements, is a hallmark of many human cancers. In 2011, advances in genome sequencing led to the discovery of something called chromothripsis, where a normal chromosome gets cut into 100 pieces, and then, in rare cases, reassembled in random order, missing out some pieces. When I saw that, I thought, how can you do that? And subsequently, we now know that in some cancers, it's really frequent. In bone cancers, it's 100% of them. And 25% of human cancers overall have a chromosome or a piece of a chromosome that has been shuffled in a chromothriptic manner. I looked at that and I thought, how can it work? And that's what got started with the work that I'm being honored for tonight. So if we fast forward 14 years, we now understand that if you just don't have, when cells divide, they've duplicated their chromosomes, and they're supposed to move half of them into one daughter cell and the other half into the other daughter cell. But sometimes one chromosome gets lost, or a piece of a chromosome gets lost, and forms its own little micronucleus, which ruptures in the subsequent phase of the next cell cycle, and it shatters. And we identified how it gets shattered, mechanisms of how the pieces can be tethered together, how they can be religated back together to make a rebuilt Frankensteinian chromosome. And indeed, it can also, little pieces can get self-ligated, just as a little chromosome piece, and it makes something called extrachromosomal DNA, a little circle of DNA which can carry oncogenes. And we now recognize that at least between 25 and 50% of human cancers have this extrachromosomal DNA. And that led the two world's largest cancer funders, the National Cancer Institute in the US and Cancer Research UK, in 2020 to issue 9 grand challenges, one of which was, how the heck do we get this extrachromosomal DNA, with 6 subheadings of the questions. And we've now given at least partial answers to each part of the 6-part grand challenge. Moreover, there was a further cancer grand challenge from those 2 funding agencies in 2015, and it was about Epstein-Barr virus, which is known to cause cancer, and in fact is the first virus linked to human cancer in the mid-1960s. And what we showed was that it encodes a protein that can bind to the viral genome, but actually it binds to one spot on chromosome 11. And when it binds there, that chromosome breaks into 2 pieces in the very first division. And one of those pieces is a gene. The closest gene is one that's very well known in the cancer community, called MLL, but basically it's a proto-oncogene for which we now know there are 51 different fusion proteins when that little gene segment encoding it is religated in a chromothriptic step to a different piece of the chromosome, a different chromosome. And so we argue that we've at least made progress on a second of the grand challenges. So my thanks to the Hope Funds for their support of 2 of my postdoctoral fellows who were instrumental in each of those discoveries that I just described. And thanks to Bill for the introduction. Yes, I grew up in southern New Mexico. My parents always encouraged my interest in science as far back as I can think. My graduate mentor, Mark Kirshner, now at Harvard, showed me how science is really done. The Leibniz Institute for Cancer Research supported me for many years. And my wife of 44 years supported me in a different way, putting up with my pursuit of a career that I love. And so finally, for scientists wearing white tie and tails, as we are tonight, is something that is really restricted to the most elite of events. The Nobel Prize is the only time you can imagine wearing that, or at the Hope Fund for, and for my introducer, Bill Kalin, it's just another night. For the rest of us, it's an amazing honor. Thank you very much.